I realized when I was reviewing that we maybe didn't have really an understanding of linked genes, and that kind of bothers me a little bit because I know we did a lab about linked genes where we did an online simulator where we were crossing fruit fly genetics and trying to figure out, what they were, what your genetics were. But I don't think that we got the gist of what linked genes were. So after reviewing, I wanted to take a little bit of time to just really discuss what's going on here. So in linked genes, let's just take an example of like fruit flies. Okay, so let's take an example like we did for our lab. And let's say that we have vestigial wings. Oh, okay, let's just get a pen, shall we? So we have vestigial wings, okay, and wild type wings. Okay, so wild type versus vestigial. We know that wild type is usually the dominant and vestigial is usually the recessive unless we're told differently. Okay, so then let me erase them. I'm trying to figure out how to use my little tools here. This is not my typical device. Okay, so we have wild type versus vestigial, and then we have wild type versus black body. Okay, so these are our two traits that we're using, and we're using a little fruit fly. So here he is. He's so cute. Okay, there's his little wings. I can't even tell what that is. Here's his little legs. Don't judge me. I'm not an artist. Okay, so the thing is that when we start with these, we need to go all the way back to the P generation. Now, they may ask you these questions in different ways, but you are going to assume the following. You are going to assume that in the P generation, you had a homozygous dominant crossed with a homozygous recessive. Oh, Miss Wilhoit, get a grip, would you please? Okay, that one parent was homozygous dominant, so he had he had wild type, so brown body, I guess, and wings, where the this is a mild disaster, where the other parent had like a black body and vestigial wings, so like that. Okay, and this is important for linked genes that you kind of think about this, because what's going on is that you have a parent, and I'm going to draw some little chromosomes here. You have a parent, and so this parent has 2 chromosomes for number 2. Let's just say this is chromosome number 2 for our fruit flies. Remember, they only have like 3. So, and then on the genes here, this purple one here, that is for body. And let's say the purple is for black body. And, oh, it's fine. And then the purple is also, but we'll make it bigger, is for the wild type body. And then on that same chromosome is your wings. Okay. And so this is important. So the thinner line is going to be our recessive, and the thicker line is going to be our dominant. Okay. So if they have babies, you're going to have babies are going to all be that, right? But here's the part that we need to talk about right here. So they have 2 chromosomes as well. All the babies do. But one of their chromosomes has the dominant and one has the recessive, right? Because they are heterozygous. So dominant, recessive. Okay. Because this one came from this parent, right? And this one came from this parent. So you are seeing that because of they are linked, that those 2 alleles for that trait travel together. So we aren't seeing like an independent assortment. If you were talking like back with, so if you had these genes were on separate chromosomes, so chromosome 2 and chromosome 3, and here's chromosome 2 and chromosome 3. Okay, so if you were seeing that, then I mostly don't know what's going on with this device. Then you'd say, okay, well, they had dominant and recessive. So, one from each parent. And since they're homozygous, then we know that they had, both the same dominant and recessive. But when this F1 has kids, like this, when this F1 has another generation, These particular ones can be assorted, like they could get this one with this one, like in meiosis, like when they made gametes, and then those two gametes came together, like they could get, what did I just do? Oh wait, let's try undo. Oh yeah, awesome. They could get the dominant, they could get the dominant wings and the recessive body color, or whatever those stand for. So basically you could mix them up, but with linked genes, they don't mix up. Okay. So you can't just do like a Punnett square, because when we do a Punnett square, basically what we're saying is, we did two little Punnett squares to calculate probability, and we'd say like, okay, they have one of those and one of those. And then their other parent, if we're crossing it with whatever. So let's say we cross it with a totally recessive, like they have that. Okay. So then this one has one of those, and then with a total recessive. Okay, cool. Because they assort independently, if they did, like we're talking if it was this scenario over here, because they sort independently, then you could say, well, they could have, there's a 2/4 chance that they'll have that and a 2/4 chance that they'll have that. So, it must be a 4 to 4 to 4 to 4 ratio or whatever. Okay. That's not how it is with linked genes. So whenever you're going to link genes, you have to be like, okay, none of that exists anymore because we are not on separate chromosomes. What we have now is, this is kind of fun, I'm not going to lie. What we have now is a situation where when you have your F2 form, so say we cross this with a total recessive, or actually let's just cross two together. Let's do a total, like a true F2. And so we cross two of these F1s together, and what did we get? Well, if it was Mendelian, then we'd expect to get a 93 to 3:1 ratio, right? But since it's linked, they can either get the dominant chromosome or the recessive chromosome. They can't get a combination of dominant and recessive unless there has been a crossing over event, okay, where when this particular individual, when they did meiosis, remember, okay, when meiosis happened in their cells, don't forget. So they started with at the top, they started with these two available chromosomes, right? This was what they got. They got the dominant from dad and the recessive from mom. We know that because that's what they had up here. They were totally dominant, like they were homozygous dominant or homozygous recessive. So they didn't have any sort of combination. So whenever those were inherited by this F1 individual right here, then what we see is in meiosis, they could have the dominant. So this is their chromosomes 2, right? And so then they make a copy of themselves, like these little tetrad things that we did in class. Okay. If they crossed over, so if you had a crossing over event between those two chromosomes, where you had, so basically I'm going to draw them as sticks because it's a lot easier. So you had your two chromosomes, and then they replicated in, your little S phase. So if they crossed over right here, so say they cut off the red one, and so then you could have a crossing over event where you could have a recombination. And so then when you came down here and you split them, and then you split them again, now all of a sudden you may have a gamete right here in the middle. These two in the middle, let's just say, may have actually swapped. And now there's a dominant with a recessive. Otherwise, if they don't swap like that, then that would have never happened. You would still, so if you didn't have any crossing over happen, you would still have these over here on the right, pick a different color, blue. So you'd still have these particular chromosome arrangements right here. Either the offspring are dominant for both traits or they are recessive for both traits, but they're not going to be recombined. You're not going to see a dominant for the wings, recessive for the body, or vice versa, because of because their parents only had a dominant chromosome or a recessive. And that's why whenever we do our F2 chart, remember whenever we say, okay, what did the offspring have? So we're drawing this little chart and we're saying, okay, so it's ridiculously small. I'm just going to erase that. Okay, so that's why whenever we see what the offspring have, what we can see, and I'll just put it over here on the left-hand side, what we can see is a situation where, so we had our wild type, black, let's just do, what did I do first? Yeah, wings. So black body. Then we have vestigial, wild type, and then we have vestigial black. Okay, so those are our options. So all of a sudden we see a lot of these, we see very few of these, and we see a lot of these. Why? Well, because their parent here, remember we crossed two of these together, so they had the same parentage, either had a dominant gene or recessive gene. And these are the ones where we actually saw a little bit of crossing over. Now, actually, sometimes you will actually see this number will be a little bit smaller because it is recessive. So if they got one chromosome from one parent that was the dominant, it would like win out. Okay. So that's kind of how, I hope that makes sense. That's why we see that these genes travel together, is because they are actually literally on the same chromosome. And so since you had parents back here that were homozygous dominant, you know that they had to have only dominant on their chromosome. And then you had parents that were homozygous recessive, you know, they only had recessive. So that's what they got from those parents. Then we crossed two of them together. You're still seeing those stay together unless there's crossing over occurs, okay? So you can either pause this now, or I wanted to make something else clear as well. And this is the law of dominance, which I didn't. These are all things that came up whenever people asked questions in class on Friday. They were things that I didn't really know that we didn't understand. And it's good. I'm glad that we had that come up because it's important. Okay, so the law of dominance. Basically, this is what dominant and recessive means. And so I'm just going to take a second and maybe talk to you more than I draw. So apologize if you just have to listen. So what dominant and recessive means is that whenever you have a gene, so, you know, this is your DNA, right? So you have two of them. So here's chromosome number 1 from mom, and here's chromosome number 1 from dad, right? And you might have, maybe you have some repeats. So then you have like that. So if you have some repeats all of a sudden out of nowhere, so instead of CGG, then all of a sudden you have a GGG, like where did that even come from? And then you have your GGA, say. I don't know. Anyway, so you have, the reason I'm saying this is because this is like your paternal DNA and this is your maternal DNA, and they're different. So this person is heterozygous for the trait, right? If they both had the ACT, this thing up here, then they would be like, if it's dominant, then they'd be dominant, homozygous dominant. If both of their genes have this long thing with extra repeats, then they'd be homozygous recessive. But our person in our example here has one of each, and so they are heterozygous. Okay, so what happens inside of your cells is whenever the RNA polymerase comes along, it codes like it has the promoter region here, and they're the same promoter region, and so the polymerase will code both of these. It might be different for, you know, the time. It might code this one, might code the paternal one time and the maternal, but they're both coded. The law of dominance does not say the dominant gene is the one that is actually coded out to protein. It says that basically when these are coded, so then from this you get your mRNA, and that goes to the ribosomes, and it makes proteins, right? Well, where we see dominance is which of the proteins do we actually see the effect of in the phenotype. So if you're talking about a disorder, like maybe this extra stuff in it, maybe it codes for a protein that starts blocking things up. Maybe it's a really large protein. I don't know. Maybe it's a large protein that like starts to block up your blood vessels, or starts to block up, you know, around and you know, just make a mess. And so basically what we want to know is, if you only need one copy of this to see the effect of it, then it's dominant. Because if you have one copy of it, you'll see the effects of it. But if, on the other hand, say it's not a large protein that blocks stuff up, say it's a protein that just doesn't work, say it's supposed to be like a channel protein and it doesn't move stuff out, like cystic fibrosis. So say it's a channel protein and it doesn't form its channel correctly, and so salts can't get out. Well, does the one that works up here, does the one you got from your dad, the one that works, does it do a good enough job that you don't even notice the difference? If it does, then that's fine. Like, yeah, you have one copy of a bad protein. Yeah, it's making bad proteins all your whole life long. But since you have a good protein up here that's actually doing the work, you don't even notice that you have a bad one. Okay, so that would mean that this bad channel protein is recessive, because you'd have to have two of them to see the effects of it. Because if you have one, then it's good enough. So that's where the dominant law comes in. It's not which of the genes is coded. They're all coded. It's which of the proteins do you see the effect of? Do you need two proteins to see the effect? If you do you need both copies to be transcribed to see the effect of it? Do you need both copies to have the bad gene to see the effect of it? If you need both copies, that's recessive. Because if you have one working one, then you won't see the effects of it. It'll be covered up. The one that's working will be quote unquote dominant. And so that's what dominant and recessive means. Dominant and recessive doesn't mean that the body chooses which one to code. It means that the expression of the gene, it's really talking about the expression of the gene. Like, if you only need one to see the effects, then that's a dominant gene. If you need both to see the effects, then that's a recessive gene. Okay. I don't know. I hope that helps, because I noticed that we had a few issues. So let me know if you have questions about that. I'll be checking my email on Monday, so that if you need any help with any of your studying stuff, then I can help you with that. Okay. All right. I'll see you later.